We're going to discuss morpholinos, which are essentially modified oligonucleotides that are effectively able to knock down expression of a gene of interest. And so morpholinos have a distinct structure as compared to most oligos. And in a morpholino, we have changed the ribose or deoxyribose ring to a six-membered morpholine ring. We've also altered the phosphodiester bond that holds those rings together. The reason that we've altered both the ring structure and the phosphodiester bond is to disable an organism from cleaving up and getting rid of this morpholino when it is injected. So morpholino is going to knock down the expression of a gene via blocking translation. And so you can have a morpholino that is complementary either to a start site or a splice site. So on the left, you'll see that a morpholino can be complementary to a start site, the protein start site. Morpholinos are 25 base pairs in length, and in the start site model, that will sit over the AUG region. Once it binds, that mRNA can travel to the cytoplasm, but it cannot interact with the ribosome. So ribosomes cannot recognize and bind with double-stranded RNA. And in essence, then that RNA is degraded and protein translation is blocked. Alternatively, morpholinos can be designed against splice sites. In this case, a splice site is going to be an intron-exon junction. So by sitting and making a double-stranded RNA at the splice site, the spliceosome, which is going to change pre-mRNA into mature RNA by cleaving out introns, is unable to do it when the morpholino is there. If that mRNA makes it out of the nucleus and it interacts with the ribosome, either you're going to get truncated protein or no protein at all, effectively blocking translation. And so we often use morpholinos in developing organisms to do a transient knockdown, which means that the morpholino cannot knock down the expression of a gene throughout the entire organism's life history, but it can do so in a short time period. And in zebrafish, this is about 4 days. And so in zebrafish, how we inject morpholino is through a glass needle, and we inject the morpholino as a liquid into the yolk of the organism as shown up top. Once the morpholino is in the yolk of the zebrafish, it can then migrate to the animal cell, which is shown here as sort of the quarter moon or half moon on top of that yolk. But it's really hard to know whether or not the morpholino has incorporated by using just light microscopy. And so we use fluorescent microscopy in order to look at the incorporation of that morpholino and hence the even distribution of this morpholino into all the cells that we're hoping to block the translation of. And so in this case, we're going to actually put a dye on the end of the 3' end of our morpholino and be able to use fluorescence microscopy in order to visualize the presence of that dye. And that dye is actually green in nature. It looks very similar to green fluorescent protein, but it's actually a very, very small dye. And when you incorporate it into the animal, that animal will look green when excited by a particular wavelength of light. In this case, we know that our gene of interest, let's say gene A, has blocked translation of gene A protein in all those cells, hence making a true knockdown.